Hello friends, welcome back to my YouTube channel Doctorscorner. Today we are going to discuss about the properties of the synapse. It is the part 2 of the synapse. Okay, in the part 1 we have discussed about the definition of the synapse, types of the synapse, structure of a chemical synapse, and how a synaptic transmission takes place in a chemical synapse, and also various types of inhibition, synaptic inhibitions. Okay, so this is the concluding part of the topic synapse. This is according to the new NMC syllabus, that is the CBME, competency based medical education system proposed by the National Medical Commission. So accordingly it is PY 10.2. okay, in the subject physiology, it's one of the competencies synapse. Okay, so we are going to discuss this briefly, because if I tell in detail again, the video length will be too long. So as I got many feedbacks that to have a shorter duration video, so I'll try to make it brief, but still it may take some time to explain few of the concepts. So let's start it. So one by one we'll discuss. The first property of the synapse is one-way conduction. One-way conduction. So the synaptic transmission, especially in a chemical synapse, okay, consider this as a presynaptic neuron, that is a neuron A. Okay, so it will conduct or transmit its signal to a neuron B. Okay, to a neuron B. So usually we have discussed in the last class how the transmission takes place at a synapse, that is due to the release of a chemical neurotransmitter in a chemical synapse. Okay, so it is usually in one direction, because the neurotransmitter is released at the neuronal terminal endings to the postsynaptic membrane, and it will stimulate the receptors on the postsynaptic membrane. Okay, so that's why conduction is only unidirectional in a chemical synapse. However, in an electrical synapse it can be bidirectional too. Okay, we are discussing about the chemical synapse, and this one-way conduction, it is also known as a Bell-Magende law. Okay, or synaptic transmission. Then the second property what we'll discuss is synaptic delay. Synaptic delay. So synapse has the property of delaying the impulse. So what do you mean by delay? That is the action potential, that is the electrical energy here from one neuron is basically transmitted to the another neuronal cell, right? That is the electrical energy is transmitted to the electrical energy of the next neuron, but in between there is a chemical energy, that is electrical energy is converted into chemical energy in the form of neurotransmitter release, and that chemical energy is further converted into electrical energy, that is formation of action potential. Okay, right? So because of this process of transmission of energy from one neuron to another neuron at the synaptic terminal, there is some delay in transmission of the impulse, and that delay is known as synaptic delay. It is just in milliseconds. Okay, and why there is a delay? For that process synaptic transmission to take place, that is release of the neurotransmitter from the vesicles, and it should bind to the receptors on the postsynaptic membrane, and it should open the ion channel, sodium, calcium, whatever it may be, and ion channel should generate the local potential, then they should get submitted to form the action potential, which we have discussed in the last class. So that time duration is that taking place for the synaptic transmission causes the synaptic delay. Okay, then we have the property of summation. So property of summation. So again, this summation can be of two types: temporal summation and spatial summation. Temporal and spatial summation. Summate means just an algebraic sum. Spatial summation. So generally, if suppose this is a one neuron A, this is another neuron B, and the impulse or the electrical energy, that is the action potential, is transmitted from this neuron to the another neuron via the synapse, right? So initially, as we have discussed, that there will be just a local potential generated, not the action potential, just a minimal amount of the current, like EPSP, around 8 millivolt current is transmitted, right? Okay, but that 8 millivolt current is not sufficient to excite this next neuron. Okay, so if we repeatedly stimulate this terminal, okay, we repeatedly stimulate it twice, then thrice or 4 or 5 times, then this will get algebraic sum, and 8 plus 8 plus 8 plus 8. Okay, for example, if it requires just 20 millivolt to get excited, and the neuronal terminal here, neurotransmitter, is just releasing an 8 millivolt energy. If that same neuronal terminal is repeated thrice, then 8 plus 8 plus 8, that it gets summated. This is known as temporal summation. It is repeated stimulation of that neuronal endings that will get summated, and that total potential what will get will now causes the action potential in the next nerve. That is the temporal summation. Then spatial summation. So again, it's also an algebraic sum. So instead of that local small potentials here, so if the postsynaptic neuron, suppose again here it is 8 millivolt, it has multiple terminals here, and it will stimulate with that smaller current here in the multiple sides of the postsynaptic neurons. Again, this will also get add up. This type of summation, it is known as spatial summation. Okay, so instead of repeated stimulation, multiple sites will stimulate the postsynaptic neuron, that will causes the spatial summation. Okay, so synapse has this property of summation. Then we have convergence. Synapse has a property of convergence. What is this convergence? Convergence, then opposite is divergence too, we see in this synapse. So this is a presynaptic neuron and postsynaptic neuron, right? So the impulse has to transmit it from one neuron to the another neuron. So there may be many presynaptic neuron or the efferent neuron which can converge on a single postsynaptic neurons. Okay, you can see this in anterior horn cells of the spinal cord. It receives signals from the spinothalamic, from the corticospinal tract or rubrospinal tract or from different tracts. So one postsynaptic neuron can receive signals from the multiple presynaptic neuron, or in the other words, presynaptic neurons will converge to a single neuron. This property of convergence of the signals to a single postsynaptic neuron or the efferent neuron, it is known as convergence. Okay, now opposite of this is divergence. So in few sites of the central nervous system, we see even the divergence opposite of that. So what is this divergence? Just opposite to that. So instead of many presynaptic neuron converging on a single postsynaptic neuron, so a single presynaptic neuron has a capability to transmit its impulse to the multiple postsynaptic neurons. Okay, so it can transmit the signal to so many postsynaptic neurons. A single presynaptic neuron can transmit the signal to this also, to this and this. So this type of transmission of the signal from a single presynaptic neuron to the multiple postsynaptic neurons are known as divergence. Okay, in simple word, opposite of convergence. Okay, now very important property we are going to discuss is occlusion and subliminal fringe. Okay, so many a times this can be a 10 marks question, describe the property of synapse, or simply many a times they will ask about these two particular properties. What is occlusion, what is subliminal fringe, as a 3 marks or 5 marks or even 2 marks in few of the universities. Subliminal fringe. So what is occlusion, what is subliminal fringe? You need to understand these terms. So subliminal fringe and occlusion. Okay, so I'll describe you how exactly it works. Okay, consider this as a presynaptic neuron. First we'll describe the occlusion. Okay, consider this as a presynaptic neuron A. Okay, I'll designate it number neuron A. Okay, now when it gets stimulated, it can stimulate 6 postsynaptic neurons. Okay, or the efferent neuron. This is efferent neuron, this is efferent neuron. So if you stimulate one neuron, hypothetically it has the capacity that it can stimulate the 6 postsynaptic neuron. Okay, I'm just giving an hypothetical example. Don't consider that a sir has said means a neuron will only excite a 6 postsynaptic neuron. Just I'm giving a hypothetical example to make this concept clear of occlusion. Okay, similarly consider this one more neuron in the neighborhood here, and that also has the capacity, when stimulated alone, it can stimulate this 6 postsynaptic neurons. This red color thing is the 6 postsynaptic neurons. Okay, so the different neurons, 1, 2, 3, 4. So I'll just put in this dotted line. Okay, because it is in neighborhood. So this 6 postsynaptic neurons. Okay, So this 6 by this neuronal A and this neuron B also 6 postsynaptic neurons. That means if we stimulate separately, the net effect is 6 stimulation of 6 postsynaptic neuron, and if we stimulate the B, that is, it is the net effect is again it will stimulate the 6 postsynaptic neuron. But if we stimulate both the neurons together, that is A plus B here, okay, unlike the mathematical formula here in the medical field, in synapses of the human body, we don't get that exact formulas here. So if you stimulate it together, what do you expect? How many postsynaptic neurons should be stimulated? If you stimulate A, will 6 postsynaptic neurons are stimulated. I am giving a hypothetical example. B also 6 postsynaptic neurons are stimulated. But if you stimulate this both, how much? It should be 6 plus 6, 12. No. So in few instances, what will happen? It will stimulate only 10 or say 9 postsynaptic neurons. Then that property is known as occlusion. Why means because these two neurons are common here in the neighborhood, okay. So this, when stimulate, it can stimulate 6 separately, it also stimulates this 6, okay, but these two neurons are the common neurons here. So this common neurons get stimulus from this A also, B also. So if they get stimulus from both also, all or none property, it will stimulate only once, okay. So this is known as phenomenon of occlusion. So instead of this separately, that is, if we have to define what is occlusion, so when an efferent neuron is stimulated and when two efferent neurons are stimulated separately, then the net result what we get is more than the sum of the two efferent neurons when stimulated simultaneously, okay. Did you understand? So when the two efferent neurons are stimulated simultaneously, then the net result will be less than the compared to the individual sum, that is individually when they are stimulated, okay. That means the occlusion, okay. Now the opposite effect here is subliminal fringe. So in few sides we see the opposite effect here, subliminal fringe. Subliminal means less than the minimum, okay, that is sub-threshold. Fringe means border, literal meaning is border, okay. So sub-threshold border. So what exactly it means? Again, I'll give you a hypothetical example. A neuron A here, again it can excite 5, okay. Here in this example we'll take, it can excite the 5 postsynaptic or efferent neurons, okay. This is efferent neuron, this is efferent neuron in the red color. So similarly B also, if you stimulate separately, it can again excite the 5 neurons, okay, 5 neurons separately, okay, that is the 5 efferent neurons it can stimulate. So this is 1, 2, 3, 4, 5. So individually, if we stimulate the neuronal A, it will stimulate 5 neurons, and if you stimulate B, then it will stimulate the 5 efferent neurons, okay. These are efferent neurons, AFFERENT efferent neurons, these are efferent neurons, okay, individually. But what happens is subliminal fringe, that is border area actually. So when it either A or B, it has some few neurons here neighborhood that is not stimulated, that is action potential is not generated, but some amount of the current passes through that neurons, okay, say here for example this A neuron can stimulate 7 or 8 neurons, but ultimately the result is seen only in 5. That 3 neurons are in the subliminal fringe, that is, for example, to excite a particular postsynaptic neuron, if the current required is 20 millivolt, say just a arbitrary value 20 millivolt, okay. So it will give current of voltage 20 millivolt to 5 neurons, but remaining 2 or 3 neurons it will give current of just 10 millivolt, okay, just arbitrarily. Similar thing happens with this neuron B, okay. Say it gives for 5 neurons 20 millivolt, and for 2 or 3 neurons it will give just a minimal current of 10 millivolt. So 10 millivolt current is not the threshold current to excite the neuron, so that neuron is not excited when they are stimulated separately. But if you stimulate it simultaneously, A plus B here, if you stimulate it simultaneously, what will happen is these neurons which are in the subliminal fringe, okay, in the neighborhood, okay, so it will get current of 10 millivolt from this A, and it will get a current of 10 millivolt from this B. So together they will get sum up, that is the spatial summation what we have discussed, spatial summation at the synapse, and the action potential is generated. So if we see here, A plus B will be 5 plus 5, 10, no, it will be more than that, here it will be 12. Okay, just opposite to occlusion. The few sites, due to this phenomenon of subliminal fringe, that is these neurons which are excited but didn't have an action potential, it is in the subliminal fringe, that is in the border area, it is under the subthreshold current. Okay, so but when they are together, they are stimulated neighboring neurons, then this subliminal fringe, they will get added or summated, and the net effect will be more. So instead of 5 plus 5, it will be 12. That is the opposite effect of occlusion. Okay, it's a bit a tricky thing, but it's such things are there. Okay, now what's the significance? We have said you some opposite things, the subliminal fringe, occlusion, divergence, convergence, summation. What is the significance here, clinical significance? I'll just give you one example why we need to study all these divergence, subliminal fringe and occlusion and all. So you know myocardial infarction or heart attack, right? So in that, basically in what happens in myocardial infarction, there is a necrosis of the myocardium, of that is the cardiac muscle, right? So the muscle dies here, that is the heart muscle or the myocardium. The pain should be felt here in the chest region, that is the precaudal region, right? So majority of the time it will be felt like that, but sometimes the pain of this myocardium is felt on the left shoulder, that is known as referred pain. Okay, why so? Again, there are so many theories of referred pain, and we'll discuss when we discuss about the pain or the referred pain. Timing, remember, is that is one of the things is because of all these properties. That is the connection, neuronal connection, the nerve supply are the commonly here, and they will, the impulse are diverted here. One neuron which is stimulated, that will get in subliminal fringe, or something will happen or summated and send the signal to the another neurons, and the another neuron will respond, and the potential is generated and the pain is felt in the other side. That is one of the clinical significance, just to create an interest to you people. Okay, so that's what we know. We need to know about these properties of the synapses in our neuronal junctions, that is in the neurons. Okay, there are so many properties, but few are important we have discussed here. Okay, now there are again more, we'll discuss again few more important properties. Then we have the synaptic fatigue. So synapse can also be fatigue like the muscles. Okay, in the neuromuscular junction, we have seen in the nerve muscle physiology, we have studied that there will be fatigue of the muscles. Okay, you might have done the mosasaurograph experiment in your physiology lab to demonstrate the site of fatigue and the phenomenon of fatigue and all. Okay, similarly synapse means the junction between the two neurons. Okay, that is the one neuron and connection or the functional junction between the two neurons. Okay, the second junction. So this junction between the two neurons, that is the neurotransmitter, is released, and if you stimulate it many a times or repeatedly, what happens is the conduction from this synapse is temporarily, it is stopped. That is, the synapse will undergo fatigue. Why it will undergo fatigue? Because the neurotransmitters which are released here, they are depleted. For the time will take for the new neurotransmitter to be produced. Okay, generally they are stored in these synaptic vesicles. Okay, and they are released, pre-stored. But when you repeatedly you stimulate it, then there should be some time for the new neurotransmitter vesicles to get generated or produced. So because of that, it is a temporary phenomenon. We see the synaptic fatigue. Okay, then the next property is synaptic plasticity. Synaptic plasticity. Again, it's a higher function, we'll discuss in detail later, but time being we should remember few of the points related to the synaptic plasticity. You know, the plastic material, so it is very much flexible, so ability to mold itself. So synapse has a property, it is a flexible, it can mold itself according to the situation. So few of these terminologies we should understand in synaptic plasticity is the postsynaptic potentiation. Postsynaptic potentiation. In learning and memory, we'll discuss about this plasticity. more in detail, but time being just remember few of the concept: postsynaptic potentiation, then long-term potentiation, then we have sensitization, then we have habituation, or also the summation. Okay, so these terms will describe the synaptic plasticity. But what is this postsynaptic potentiation is? So consider this again, if one neuron, okay, it's getting transmitted to the signal to the next neuron, neuronal A and neuron B. Okay, so when you stimulate it, when you stimulate this first neuron, then it will send the signal to the second neuron. When you repeatedly stimulate it, so sometimes repeatedly in a short interval time, the next stimulus will causes the increased response in the postsynaptic membrane, that is known as postsynaptic potentiation. Why it happens so? Because when you stimulate for the first time, then there is a release of one neurotransmitter here. Okay, there is a release of the one neurotransmitter here, postsynaptic or post-tetanic potentiation. More specifically, it is post-tetanic or long-term potentiation, or the long-term post-tetanic potentiation. It is post-tetanic potentiation or long-term post-tetanic potentiation. Okay, so what is this long-term or just post-tetanic potentiation, or the postsynaptic or post-tetanic potentiation? So suppose one stimulus if you give, and after that you give a repeated stimulus, okay, that is 100 per second, then again you give a normal same stimulus, then there is increased response seen here in the postsynaptic neuron, that is known as post-tetanic, that is tetanus, repeated stimulation. Okay, why there is increased response after the post-tetanic stimulation is because the calcium channels get opened up at the nerve terminals, and also the increase in the release of the neurotransmitter, which causes the increased effect in the postsynaptic membrane. Okay, that is known as post-tetanic potentiation. And the same thing if it is done for a longer time, it is known as the long-term post-tetanic potentiation seen in hippocampus and all. Then sensitization by irritant stimuli and all, and habituation. You might have seen that when there is a prick of a thorn, okay, on the sole, so initially there is a sharp pain, but after a few minutes, if the thorn is not removed and it is still on this side, then the pain is not felt as sharp as it was in the initial time. That is due to the habitualization and sensitization phenomena of synaptic plasticity. That is, the synapse has the ability to adjust according to the situation. That is known as synaptic plasticity. Okay, again there are so many terms we need to study in this. We'll study in detail while we study the learning and memory. Time being, just remember this important terminology, that is post-tetanic potentiation, sensitization, habituation and all, which is related to the properties of the synapses. Okay, then we have one more, facilitation. I think facilitation, time is going on, but still I'll try to finish it in with next 15 minutes. So facilitation, again same like post this one, there is one presynaptic neuron, then postsynaptic neuron. Okay, so if you stimulate once, then it will transmit the signal from this presynaptic neuron to the postsynaptic neuron, right? But if you stimulate it repeatedly again, then the next response is seen as an increased response, that is known as facilitation. That is, a previous stimulus will facilitate the next or succeeding stimulus. How it will stimulate the next facilitate is because again the same mechanism, release of the calcium and all. So already due to the opening of the calcium channels here, the more calcium is stored there, then that calcium will facilitate the release of the neurotransmitter early. So when the second impulse go for the second time, so it is facilitated by the previous experience. Okay, that is known as facilitation. Like same like synaptic plasticity, that is, it will learn by its experience. Okay, so this is the property of facilitation. Then we have reverberation. So reverberation, then we have, I'll just enumerate the properties first, then I'll describe it first. The reverberation, we have the reciprocal inhibition, reciprocal inhibition. Then we have after discharge after discharge, then effect of hypoxia and acidosis on synapse. Okay, so there are so many properties here. So just I'll quickly just tell what is this reverberation and reciprocal inhibition. So what is this reverberation? So reverberation means, suppose this is a presynaptic neuron, okay, neuronal A, okay, neuron A, and another neuron that is postsynaptic neuron, neuron B, right? So in reverberation, what happens when a signal from this neuron A goes to this neuron B, this neuron B, this axon may sometimes, they will have one process that will excite the neuron A once again. So that is, neuron A will causes its own excitation once again by the reverberation circuit, that is known as one of the properties reverberation, or need not necessary that it should give directly through a different neuron. Even this one of the process of the axon may, it will stimulate its own dendrite, it is again it will get stimulated by its own, that is known as reverberation. Okay, then what is reciprocal inhibition? Again, one of the very important property, very useful in synapse, this one reflex phenomena. You know, different reflexes. So in your physiology practicals, you might have studied the biceps reflex, triceps reflex, so many tendon reflexes you will be doing, right? So generally in a reflex, so one of the muscles, like for example flexor muscles are contracting, the other muscles are relaxed. So how they are kept relaxed, okay, how they are kept relaxed is, so I'll just explain you. So I'll just show a cut section of a spinal cord here. So this is dorsal root and this is the anterior horns. So in a spinal cord, a efferent neuron from this dorsal root come, and here it will synapse with an efferent neuron. Okay, it's with an efferent neuron. So it will give connection, to two groups of muscles, flexor muscles and extensor muscles. Okay, it will give connection to the flexors and the extensor muscles. Okay, with the help of this green pen I am just drawing this. These are considered as the flexor muscles and these are considered as the extensor muscles. Okay, now the signal from this efferent nerve will synapse at this cell, that is the anterior horn cells here. So it will stimulate the one group of muscles, okay, that is the flexor muscles. It will stimulate the flexor muscles. Simultaneously it will stimulate one more interneuron in between, that is one more interneuron in between, that is an inhibitory interneuron. Okay, inhibitory interneuron that will inhibit the extensor group of muscles. Okay, so it will stimulate the inhibitory interneuron which will opposes this extensor muscles. This type of property is known as reciprocal inhibition. That is, one group of muscles is stimulated and simultaneously when they are stimulated, this extensor group, that is flexors are when stimulated, extensors are inhibited. So this is known as reciprocal inhibition. Then after discharge, and this reciprocal inhibition is due to the inhibitory interneuron. Okay, this efferent neuron will stimulate the flexor group of muscles and causes the stimulation of inhibitory interneuron. This inhibitory interneuron will stimulate the will cause inhibition of this extensor group of muscles. After discharge means whenever there is a synaptic transmission, there is some amount of some discharge is remaining, known as after discharge. Then synapses are very much sensitive to hypoxia and acidosis, that is changes in the pH and all. So whenever there is hypoxia and all, so there is synaptic transmission is affected. So because of this synaptic, even this fatigue, so the most common site of the fatigue is the central nervous system because of this hypoxia and acidosis. Okay, so these are the few properties of this synapse. So just to summarize, and I'll just enumerate this thing: one-way conduction, synaptic delay, summation, either temporal or spatial summation, convergence, divergence, occlusion, subliminal fringe, synaptic fatigue, synaptic plasticity, that is post-tetanic potentiation or long-term long-term tetanic potentiation, then sensitization, habituation and summation here, then facilitation, reverberation, reciprocal inhibition after discharge, and effect of hypoxia and acidosis. Okay, thank you very much.